in the area of adaptation and what I call carryover designs. So that's going to be the main focus today. The thing that we're going to cover is different types of neural coding, so how fMRI can be used to measure different sorts of responses to stimuli and how those different sorts of neural codes can tell you about object properties. And we'll concentrate in particular upon one technique called adaptation. And look in particular how a property of that adaptation measure can inform as to sort of the shape of receptive fields of neurons, what their tuning is like for object properties. So that's the topic for today. And not Thursday, but on Friday, I'll talk a little bit about the relationship between adaptation measures and multivoxel pattern analysis and how the two techniques can give different information and the circumstances under which that can happen. So that's the marching orders. And then tomorrow is the seminar where I'll talk about something completely different, which has to do with the organization of the brain for vision and its relationship to anatomy. So for those who are interested in some discussion of anatomy and how MRI can be used to measure anatomy and align anatomy, that discussion will come tomorrow. Okay. So, and again, as before, happy for interruptions as we go along and questions, and we'll take a break about halfway through. Okay, so what I wanted to introduce was this idea that there are different forms of neural coding, that information about objects or language or anything else can be represented by different patterns or forms of neural activity, and that these different forms of neural activity might be of interest just for basic theoretical reasons, but that it's important to recognize that different neuroimaging measures will be sensitive to different kinds of patterns and forms of neural activity. And so the area in which I work is in visual perception, and particularly for object properties. And I know that many in the audience are interested more in language and higher level cognitive function. So I wanted to give a quick two-slide introduction to the part of the brain that I spend my time thinking about. So this is a view of the underside, the ventral surface of the brain, after each hemisphere has been digitally inflated, right? So these are quite common representations these days of data. So what we're doing is we're looking at the brain from the underside. So this is the back here. The front up there is where the eyeballs would be. And so this is the occipital lobe, and then coming into the temporal lobe here. And so what is illustrated on this inflated view of the human brain are the location of several of these categorical visual areas that have been identified over the last 20 years. And so these are patches of cortex with the perhaps surprising property that they tend to respond more to certain categories of stimuli than to others. So some of these are quite famous. So, you know, the fusiform face area, or FFA, everyone has heard about that, I'm sure. This is a piece of cortex where the neurons respond more to pictures of faces and information about faces as compared to other sorts of stimuli. There is the parahippocampal place area in red, an area that responds to environmental stimuli, the appearance of landmarks and street scenes and buildings and rooms, so things with a spatially extended layout. And then there's the LOC, or lateral occipital complex, which is indicated in blue, and this is a more posterior region of visual cortex that responds to all sorts of visual objects, you know, shoes and ships and cabbages and all sorts of things. So all sorts of different visual formed objects produce greater activity in this region than unformed or scrambled visual objects. And there's other areas as well. The extrastriate body area that responds to body parts, the visual word form area. Some of these areas are still being defined, and are there, you know, did they come in multiple pieces or just a single region? But in general, it's the property that ventral cortex has these discrete areas that seem to respond to different kinds of stimuli. I think that's probably a familiar concept for many in the room. So that's a great first step for understanding the organization of visual cortex. But, you know, we might want to know a little bit more about how it is that neurons within these regions code for variation in the appearance of objects, right? So we can tell faces from houses, and we can tell, you know, irons from from ships or words, but we might be interested in this kind of thing, which is being able to tell apart individuals, right, and recognize and store the appearance of different exemplars within a category. And so that might require a different kind of coding than just having one piece of cortex for Tim and another piece of cortex for Barbara and another piece of cortex for somebody else. And so that's a little bit about what we're going to talk about. Okay, so generally there are these visual areas. They're specialized for different categories of visual information, but that we might want to probe a little bit more deeply about how information for variation of exemplars is stored within there. Okay. So what we're going to talk about a little bit is a type of fMRI experimental design that I call carryover designs that can be used to measure three basic types of task-evoked neural response, right? So I'm going to argue that there's three basic sorts of measures that you can get out of fMRI data in response to stimuli and tasks, and that I'm going to present a type of experiment that is sensitive to all three of these sorts of measures, that can measure all three simultaneously. So first let's talk about what those three types of measures might be. So the first kind of measure is just a measure of the straightforward amplitude of bold fMRI response to different types of stimuli, right? And so this is the most traditional and classic type of fMRI experiment. And this is the idea that if you put somebody in the scanner and you show them pictures of faces and irons and houses, you might find a piece of cortex that just responds more, has greater bold fMRI signal to the pictures of houses or faces as compared to other things, right? And maybe within primary visual cortex, you might find that if you vary a stimulus such as the contrast of a stimulus, this has high contrast and this patch has low contrast, you might find that the response within area V1, this early visual area of the brain, that it increases, that the fMRI amplitude increases with the amount of contrast. Okay, it's a pretty straightforward idea, and this is really the oldest type of fMRI experiment. You know, you vary a stimulus and you see, is there more fMRI signal, which you interpret as more neural activity, right? So I'm going to give us a term for that. We're going to call that the average direct effect of a stimulus. Okay, so across all the times that I presented the face, if I take the average of all the face stimuli I presented, the response that you get to that stimulus we're going to call the average direct effect. Okay, so that's one type of measure you can get from fMRI that's been around for a long, long time. So, you know, you can also look at that direct effect and not just take its average. You can take a look at the pattern of those responses across a set of voxels. And this is the basis of distributed pattern analysis or multi-voxel pattern analysis. This slide illustrates conceptually the kind of study that Jim Haxby did in his science paper back a few years ago that sort of brought MVPA back into sort of the awareness of the field. And so the idea here is that instead of taking the average fMRI response across a set of voxels or points in the brain, instead you make a list. You make a list of all the voxels and then the response of each voxel to one stimulus and to another stimulus. You make the whole list, and you do this for one stimulus type and another stimulus type. And then you're interested in, broadly, what's the correlation between the set of responses across voxels to one stimulus type as compared to another stimulus type. So these two faces evoke similar vectors or patterns of responses across all the voxels. So they're correlated at 0.75. And same with the chairs, they have similar patterns, and the houses have similar patterns. But the patterns between the categories are not as similar. Okay, and so this is the one type of multivoxel pattern analysis. There are other techniques where you take these vectors and you use them as information to train machine learning classifiers and support vector machines. So that's the general idea. So, but in any event, essentially what's being measured here is this is the pattern of direct effects across voxels. You have a set of voxels, and you measure, hey, how much did this point in the brain, like stimulus 1 and stimulus 2, averaged across all the times I presented it? And then you get that pattern across all the voxels. And that's the basis then of a multivoxel pattern analysis. Any questions about that basic idea? Okay. But then there's a third type of measure, or third type of way that you can measure out neural activity, and that's using something called within voxel adaptation. And this is a technique that probably was its most popular about 10 years ago, and multivoxel pattern analysis has swept into being sort of more used in recent years. But the basic idea is that there's a physiologic observation that neurons habituate. They seem to have a reduced response to the repetition of a stimulus. And this is something that DeSimone demonstrated many years ago with macaque neurons in infratemporal cortex and in many other contexts. And the model, the idea about what's happening is that if you have a patch of cortex, and so that's my cartoon for a patch of cortex with a set of neurons, and you present a face, then the neurons that represent that face fire. So they have if the neurons are tuned to like that face appearance, those neurons fire. But if you present the face again, then those neurons are broadly fatigued and respond less than they did the first time. However, if you present a face and then present a new face, then a new set of neurons now respond, and they have a release from adaptation. So they no longer are suppressed in their response. That's the basic model. But if you then take the average response across these two stimuli, which are presented close together in time, right, closer together in time than functional MRI can resolve, if you measure the average response to these two, then you'll see you get a smaller response for when you have the same pair of stimuli, and a larger response for a different pair of stimuli. This is the basic idea behind neural adaptation studies, which present pairs of stimuli, one after the other, and then vary whether or not the pair of stimuli are the same or different, or vary the similarity of the pair of stimuli. Okay? So this is unlike the other kinds of measures of neural response that I described, because here we're looking at the modulatory effect of one stimulus on the next. Okay? So we're not interested anymore in what is the average response to this guy. We're interested in what's the response to this guy depending upon how similar or dissimilar was the prior stimulus that came. Okay? So it's the modulatory effect of one stimulus on the next. So traditionally, these sorts of measures have been studied in completely separate experiments. You do an experiment that was just to look at adaptation, or you would do a different experiment just to look at multivoxel pattern analysis, or a different experiment to compare the amplitudes of response to different stimuli. And the reason that one might be interested in measuring these different kinds of neural response is because different kinds of neural coding might give you different types of answers with this kind of measure. Okay? So these different measures might be sensitive to different kinds of neural representation. So let me just walk you through a cartoon of that. So here's the idea of we have a portion of the infratemporal cortex of the brain. Maybe these are 4 voxels that like objects, and you would like to know what is it about objects and shapes that these voxels respond to. So you present pictures of apples and you present pictures of pears. So in this example, let's imagine that those 4 voxels, which I now have illustrated here, those 4 voxels corresponding to that those 4 voxels are apple voxels. They like to respond to apples more than they like to respond to any other kind of fruit. So in this example, they're receptive fields, which are indicated at the top. So that's what the neurons in that voxel like. Those receptive fields and those neurons, they fire more for the apple than they do for the pear. So the amplitude of response across each of the voxels is 1 unit of response, 1 unit of response, 1, 1, but no response to the pear. And if you just take the difference between those two, that's the classic direct effect. This is how you would find, you'd have the face area, the house area, and now you'd have the apple area, right? This would be the evidence for an apple area, that it responds more to apples than to other sorts of things. So that's one kind of coding, right? So then another kind of code, so that's a measure of the direct effect, but then another kind of code that you might encounter is this. Suppose those different voxels have tuning for different basic object properties. You have one set of neurons that like red things and one set of neurons that like yellow things. and one set of neurons that like round and another set that like long things, right? So in this case, what you would find is that different populations of neurons, so the voxels would respond differently to the apple and to the pear. And although the average response across all 4 of the voxels is the same, right? So 0 plus 1 plus 0 plus 1 divided by 4, that's, half, 2, 1. Half, that response is the same as the response here for the pear. Nonetheless, the pattern of response across the voxels is different. Okay, so this is then the across voxel pattern, and this would be a type of representation that multi-voxel pattern analysis would be sensitive to. Okay, and so then just finally, to bring this all the way in, a final type of representation might be, suppose all of these voxels contain populations of neurons which are fruit salad neurons. They code in a population manner the space of possible fruit, right? So you have, in all of these voxels, you have different neurons which are intermixed, and that some of the neurons like more like kiwis, and some like oranges, and some like apples. And so in each voxel, it's a different pattern of response across the different fruits, okay? So the apple gets one pattern of response across the voxels, and the pear gets another pattern of response. So now, the average response across all the voxels is the same between the two, and the average response within the voxel is the same between the two. So distributed pattern analysis would not show any difference here. But within voxel adaptation, using that adaptation technique, might be able to reveal this tuning, this representation of fruit space. Okay, so any questions about this sort of elementary division or simple model of different kinds of coding? Okay? So these are different things. These are different models which are implicit in many studies that have been conducted with functional MRI over the years. They're either looking at direct effects, they're looking at these distributed patterns, they're looking at adaptation, and really a model of neural tuning across voxels is implied by those different studies. Okay. So it turns out that there's a way that you could measure all 3 types of this representation with just one experiment, and that's what I call a continuous carryover design, or a carryover design, okay? So it's a way to simultaneously and efficiently measure within voxel adaptation and across voxel distributed pattern responses. And I'm going to argue that what it's really best for is doing these sorts of representational similarity analyses that Nikos Krigaskort has come up with a great name for. This idea that you're going to relate the similarity of a set of perceptual objects or conceptual things to the similarity of the neural responses that they evoke. So that's where we're going to go with this eventually. The key idea, or the key insight of the approach, is that you don't need to present your stimuli in isolated pairs to measure adaptation. Instead, you can present them as a continuous stream, bop, 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 one after the other, okay? One important feature of this kind of experiment that we're going to return to is that the order of the stimuli has to be counterbalanced, and we'll talk about why that is in a moment. Okay, so this approach, this whole set of ideas, has its root in a very different set of ideas, in the identification of good tobacco leaves and in graph theory. So let me see if I can explain this. So tobacco leaves. So tobacco leaves, you make cigarettes and cigars out of those. In the 1950s in the United States, there were these very large tobacco farms, right? So you'd have a big farm, many tobacco plants, and you'd grow the tobacco, and then people would pick the leaves, right? So now you'd have a huge pile of tobacco leaves. And the problem was that some of those tobacco leaves would have disease on them. Like the fungus would get on the tobacco leaves, and then they'd be no good. You wouldn't want to roll them up in a cigarette because it would taste bad. So there were workers that would stand at a conveyor belt, at a belt coming by, and the leaves would come by one after the other. And their job was to sort the tobacco leaves into good, not so good, and bad. Okay, so they had to rate each tobacco leaf whether it was good or bad. And the owners of the factory who had the workers doing this were worried that their response, that their judgment, if a tobacco leaf was good or bad, would depend upon the last tobacco leaf they saw. So if they saw a bad tobacco leaf, maybe the next tobacco leaf, which was bad, was still better than the last one. So they'd say, good enough, right? So if you see bad, bad, not so bad, you might say, it's good, right? So maybe your response, your judgment about whether a tobacco leaf is diseased or not depends upon or is influenced by the last tobacco leaf you saw, right? That there's an effect of context. And so this actually became a research problem for what were called industrial psychologists. So psychologists who were interested in helping tune the function of different industrial processes. And they developed techniques for solving this problem, for being able to figure out how much of an influence the last tobacco leaf had upon your rating, okay? And the solution is really simple. It's a very simple approach, and it all can be incorporated within a linear model, okay? And so this is the only equation I will show you, and it's not a scary equation. It's really easy. All it's saying is that now in the context of neural responses, the neural response, or your response equivalently to a tobacco leaf, so your neural response to a stimulus at a given position in time is going to be the result of the sum of these different things. And so those things are the overall mean response, so your response to just, on average, all the things you saw, the response to the thing itself, right? So you can imagine if you had 3 different types of tobacco leaves, bad tobacco leaf, medium tobacco leaf, good tobacco leaf, that your response to any one leaf is your mean response plus your response to good, medium, or bad, plus the influence of the prior leaf. That's all that says, that there's going to be your mean response, your response to the leaf itself, to the stimulus itself, and the modulatory effect or influence of the prior stimulus you saw, okay? And so this modulatory component is actually a matrix. It's a matrix which is the last thing you saw versus what you're currently looking at. And we'll talk about that some more in just a moment. So this effect, the effect of the stimulus itself upon your response, that's referred to as the direct effect. It's the direct effect of the stimulus. What would your response be? What would your rating be of that leaf all on its own? Okay? So independent of context, what's your response to that leaf? And the response here, this is the modulatory effect of the prior stimulus. So in terms of fMRI, this modulatory effect, that's within voxel neural adaptation. Okay? That's sort of the influence of the prior stimulus upon the current stimulus. That's adaptation. Or we'll see adaptation is a type of modulation. But then the direct effect, well, that's the basis of then doing like a distributed pattern analysis. And so we'll go through some examples of this more in a moment. Any questions about that so far? You with me? Okay. So just to kind of make this concrete, let's talk some more about what this matrix is like, okay? So imagine you had a set of 5 faces, right? 5 faces across the top. And you present those faces one after the other after the other. And you're interested, what is the response to a face as a function of how similar the prior face was, or as a function of what the face that came before it was, okay? So if you make, I'm sorry, this has more detail than it needs, but this should make the point. So this matrix is the carryover effect. And so this is the current stimulus, face 1, 2, 3, 4, 5. And this is the prior stimulus, face 1, 2, 3, 4, 5. Okay? So that matrix then describes how much of a modulatory effect. So for example, along the diagonal, this is when a face then followed itself. So a face repeated. There was an exact repeat. Okay? And the scale, the gray, indicates whether there was a reduced response or not much of a change from the mean, or an enhanced response, a response that was greater than the mean. Okay? And so this matrix captures all of the possible ways that a prior stimulus can influence the current stimulus. And in fact, as a matrix, you can then decompose this into submatrices, into a basis set. You can examine different components that make up that influence of one stimulus to the next. And I will show you just a couple of examples. So these are some of the matrices that if you add these together, you can replicate the entire carryover matrix. So just for example, one type of carryover effect, one modulatory effect of the prior stimulus upon the current stimulus might be neural adaptation. Okay, so that's one type of modulatory effect that many people are interested in. So a linear adaptation just means that the value in this matrix is proportional to how different these two stimuli are up there. So how far apart these stimuli were. Was it a small step or a big step from one stimulus to the next? And it has the property that it's symmetric, so that for neural adaptation under those models that are used, the belief is that moving from face A to B has a modulatory effect that's the same as going from face B to A. Okay, so that's a linear adaptation effect. That's one component that could be present within this entire carryover matrix. But there's other stuff too. There can be what are called bias effects, and these are actually the effects that the tobacco farmers were very worried about. This is the kind of effect where going from one stimulus to the next is different going one direction than the other direction. So going from face A to face B produces a small bias or a small change in response, but going from face B to A produces a large response. Okay? This is an asymmetric or bias effect. These kinds of effects, these bias effects, have been studied very much in the realm of what's called sequential judgments of magnitude. And so things like judgment of the contrast of stimuli or how loud a stimulus is, these bias effects are very present or very prevalent in those sorts of things. So suppose you're listening to sounds, and some sounds are very quiet and some sounds are very loud. Your response to a sound is influenced by the loudness of the prior sound in an asymmetric manner. So it's different going soft to loud from loud to soft. Okay? So that's a different modulatory effect. That's a bias effect, and so that's another type of carryover effect. Okay? Any questions about that idea? That there's this whole space of the influence of one stimulus to the next, that a matrix captures all of those influences, and that one type of influence within that matrix is linear adaptation, which is the sort of measure that fMRI studies have traditionally obtained. I know these are a lot of sort of big ideas coming fast, so please feel free to stop and ask me if you have any questions about it. No? Okay. I'm happy to stop any time if any of this is confusing. Okay, so the final piece of this is that these direct and carryover effects are estimated efficiently and without bias when you have a counterbalanced stimulus order. So, for example, if you only ever presented the stimuli da 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 da, then you could never tell what was the effect of asymmetry and symmetry, right? So you could never know what was the effect of the stimulus and what was the effect of the carryover if the stimuli always increased in order, for example. So you need to be able to have all combinations of having the quiet stimulus before the loud stimulus and the loud stimulus before the quiet one, and all possible orderings. So an ordering of stimuli that provides that kind of balance of the history is called a counterbalanced stimulus. So there's an interesting topic to discuss here about how one creates counterbalanced stimuli or counterbalanced sequences. I will just say generally, and this is something that we can discuss more on Friday if folks are interested, that there is a mathematical technique using graph theory that can be used to create sequences of items which are counterbalanced like this. So, the general idea is that if you had three types of stimuli, if you had a house and a tool and a face, and you wanted to produce an ordering of the stimuli so that you had house to face and face to house and house to tool and tool to face and tool to house and so on, that one way to create such an ordering is within the context of a graph that then you find a certain path through, and then that creates the sequence. We won't talk any more about this today, except to say that there are methods to create an ordering of stimuli that has the property that every stimulus comes before and after every other stimulus. And that's referred to as a counterbalanced stimulus. And one property of these stimuli is that these sequences is that they can be very long. So if you have, say, 5 stimuli, and you want to control the history of the stimuli going back not just for the stimulus that came 1 back, but 2 or 3 or 4 back, so control the history of the stimuli going back in time, you need quite a long sequence, okay? So this is one example sequence for if you wanted to study, say, 5 faces or 5 words, and then control the history back over time for the last 5 stimuli. One property of this sequence and of these counterbalance sequences in general is that they are useful in cryptography, for like breaking codes. So de Bruijn sequences have a role in creation of keys for digital keys for secure transmission of information and for breaking codes. And the reason is that one of these sequences is a very efficient way to try all the combinations on a lock. That's one way of thinking of it. If you had a lock and you needed to try every combination to get the lock to open, this sequence is a very efficient way of trying every combination. In fact, this sequence is just the right sequence for breaking into cars. So some American cars have, instead of a key to unlock the car, you have a push-button pad that will then open the door. So this sequence is the perfect sequence to then punch the buttons on the car and then the door will open, okay? So there's actually some deep connections between the idea of sequences which are good for code breaking and sequences which are good for measuring patterns of neural activity. Because what you're trying to do is, when you're measuring a neural code, is you want to examine the response of the neurons to every possible combination of the stimuli that you use. Just in the same way that if you want to open a lock, you need to try every possible combination of the buttons, right, on the keypad. So there's a connection between the two applications in that way, okay? And again, in a topic that I'm not going to discuss today, it's possible to create these sequences in such a way that they are very efficient or well-designed for doing functional MRI studies. It's as if you could find the most efficient way of punching the code on the lock if your hand had a cramp. That's the physical analogy. Is for fMRI, you can create an ordering of the stimuli which is very efficient for fMRI. So, okay, let's actually have an example now. So that's enough theory. Let's look at an example study so you can see how all of this would work. And so this is the example study that I used in the original paper in 2007 where we described this. So the goal is to examine the relationship between the similarity of a set of colors and the similarity of the neural responses that those colors evoke, okay? So color, can be defined as a point within a 3-dimensional space, right? So you have red, green, and blue. There are other sorts of color systems as well. This is what's called the OSA UCS color space. And this is a way of defining color so that every one step in the space is perceptually equal, roughly. So that, this stimulus and this stimulus, these two are as similar as this stimulus is to this stimulus, and so on. Okay? So it's a way of creating equally spaced colors that are equally spaced in perception. And what we wanted to know is it the case that the similarity for visual cortex of one color to another color, does the neural response reflect the similarity of the perceptual similarity of the colors? That was the goal, okay? And to do so both for neural adaptation and across voxel patterns. So, these set of colors, if you number them 1 through 27, then that implies a similarity space or matrix which describes how similar one color is to the next. So, that's the one color and the next and the next and the next. And then up here is coded how similar one color is to another color by looking at it. So how perceptually similar it is. Okay? And so the design of the experiment is you put subjects in the scanner and you present them these 27 colors over and over and over and over again. Bop, 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 bop. So the colors just flash by on the screen, one after the other after the other. It's a kind of boring experiment for the subject. So just color, 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 color. And the ordering, it seems completely random to the subject. They can't tell the difference they can't predict the ordering of the colors. So, and while they're looking at these colors, they're watching a fixation cross in the center of this screen. Okay? So their job is not to make any decisions about the colors, but just watch a fixation cross and watch in case it changes from black to white. That's what they were doing. So the colors come one after the other. So and then while these colors go, we measure fMRI data. So then here's how we measure adaptation. Yeah?
How fast were they coming?
Oh, so the stimuli in this experiment were coming one every 1,500 milliseconds. And so this kind of experiment can be done with faster or slower presentation times. We've done this with 6 seconds per stimulus, and that's an experiment that we conducted with Russell Epstein, who was here recently. He might have presented this work, to look at the similarity of representation of different places. And so that was every 6 seconds for the stimuli. And you can present them even faster. We've done this with the stimuli coming every 1,100 milliseconds in another color experiment that I won't discuss. So yeah, so they come fairly rapidly. So bap, 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 yeah.
Yeah, sorry, George, you told in the presentation you put all the colors, the possible colors, the 27. Yeah, and then in the within sequence, you repeat some values.
Yes, the colors get repeated over and over and over again. That's right. In fact, each color is presented 27 times. Okay? And that's necessary because what you want to measure is what is the response to this color of violet when it is preceded by, when it comes after, blue and orange and green and yellow and right? So you examine what is the response to a color after every other color. And so to do that, you need to present it 27 times. Okay? So it's a lot of colors. They come on by. Okay? Okay.
So do you have just one measure?
What's that?
Do you have only one measure? If each color is repeated only once, you have only once for each pair.
Oh, once for each pair. That's right. That's right. So, or instead of pair, maybe each transition. That's right. So each transition is only measured once. That's right. If you just went through the sequence one time, but we went through the sequence 3 times, or twice. We went through a couple times. Yes, that's right. So each transition is only measured one time through the cycle. Okay. So here's how these data are analyzed, and here's what you go looking for. Okay. So up here on the right, this is how perceptually similar any one color is to another, right? Okay, so blue is very similar to another shade of blue, but dark green is very different from orange. And so this matrix up here shows how similar one color is to another. Okay? And down here, this is the order of the colors across the scan for one little piece of it. Okay? And what we want to create is this predictor, this covariate, that is going to predict how much of a neural response there would be to a stimulus depending upon how similar it was to the prior stimulus. Okay? So let me see if I can illustrate that. So here at this point, there was a transition between one shade of green and a different shade of green. Okay? Those two colors are quite similar, right? So their perceptual similarity is quite high. They look the same. Okay? So we would predict that the neural response, the modulatory response, would be a very small response. You would tend to have a small response to the color because it's quite similar to the one before it. under this model of neural adaptation, right? So the neurons that like that shade of green would also be relatively adapted by a similar shade of green, okay? But then there's another transition, which you might show here, which is a medium change that's between pink and brown. And so that predicts then that there would be a slightly larger neural response or a slightly larger recovery from adaptation. And then finally, you might have a very big change from the light green to a purple. And so you'd predict a big modulatory response, right? A big recovery from adaptation. And so you can do this just sort of marching along the sequence and at each point in time predict what is the response to the stimulus as a function of the prior response, okay? And an additional nuance of this is that we can do it not just for a linear effective similarity, but for higher order components as well, quadratic and cubic, but let's leave that alone for the moment. But it's possible to model not just a simple linear relationship, but to expand that with a basis set into more complicated relationships. Okay, so these are just the results from one subject who we scanned with this experiment. And what I'm first showing you is, and I didn't mention, in that experiment as well, there's also blank trials when the screen is just black. Okay? So first, this shows the response for seeing any color versus the black. And so this is that mean response. You remember the first component of the equation, the mean response to all stimuli. So what you can see is that this is now back in the visual cortex. You're seeing it on the medial view. That's the lateral view. And there's multiple visual areas that appropriately respond to this patch of color just flashing in the center of the screen. Okay? V1, V2, V3, HV4. We can find each of these visual areas. These are early retinotopic visual areas for those who are not familiar with that terminology. Okay, so that effect of a stimulus versus the blank screen, very powerful, just as you would hope. But then what we can do is we can create a plot, which is how big was the fMRI response as a function of the change in color space, in the size of the change in color, okay? So for example, down here, this is where the color was exactly the same from one trial to the next trial. And this is where there was a small change and a medium change and a large change in the color. And so within these early visual areas, the neural response, the fMRI signal, scaled roughly linearly with the size of the change in color. So this would support a model of a population code of neurons that represent the colors, and that there's adaptation that's proportional to the similarity of the colors. So let me stop there for a moment. Does that make sense to everybody? This is a measurement of adaptation which is parametric. You're measuring sort of bigger adaptation and smaller adaptation depending upon how dissimilar the colors are. Okay? So that's one type of carryover effect. The effect of moving from, orange or peach to green is the same. We predict as the model as the same as green to peach. That's that symmetric adaptation effect. Okay. Oh, and by the way, that's right. You asked about, we only get one instance of each pair, of each transition. Actually, you kind of have two for this type of model because you have peach to green and green to peach. So you actually have, you put together for this modeling each direction of the transition. You count this.
But you also have the same changes not from the same pair of colors, but two colors which are different.
That's right, but have the same dissimilarity. Exactly, that's right. So you have many pairs which are step size 1 and many pairs which are step size 2 and so on. Okay, so that's one type of carryover effect that might be measured within this. But there's another type too, and this is the idea of bias. So this is this notion. Why, do you have a question?
I thought that maybe you have more one-step changes.
That's
compared to 2 and 3. And
that's right, exactly. That's exactly right.
More powerful for the one-step similarity compared to the 4.
That's right. So this is an important point, is that the number of transitions of a given size is different for the different size transitions. So there's only a few ways that you can make the biggest change in the space, but there's many ways you can make the small change. So that's right. You have more trials to estimate small changes than big changes. And that's a necessary feature of having the balance of the stimuli. Tomorrow, we'll talk a little bit about how that effect of having different numbers of transitions can lead to, has produced in the literature a number of papers with wrong claims about a type of representation of object similarity. And so you need to correct for that kind of thing, but we won't talk about that today.
I mean, the variance of the curve is different on the top part.
That's right.
You plot the average, you don't see that, but if you plot the...
That's right. That's right. You'll see, so this was the data from one subject. You'll see data from additional experiments in a bit where I have error bars across the transitions. You'll see, yes, the error bars get larger as you move to bigger transitions.
Yes, you can control for that. You're presenting more times the ones that have a higher distance.
Okay, so the point that's being made is, hey, maybe you could create a sequence which is enriched for the large transitions. That's true, but then it would no longer be counterbalanced, right? So you can't do both things at once. So tomorrow I'll discuss a way in which you can model that effect and try and remove it to avoid a confound, which I won't discuss today, but that can appear tomorrow. And it turns out that just in studies of, just even in macaque studies of single units, this effect of having different numbers of the sizes of transitions has, in my opinion, affected the claims that have been made about the types of neural coding that's present. How neurons represent, for example, the appearance of faces. But we'll come to that tomorrow. So in any event, the one other type of... Oh, yes?
Sorry,
sure.
If you go back to the previous slide with the data.
Okay.
Given that they are in fact different colors, why do you get adaptation at all? I mean, I can understand the relationship between the size of the difference between the two responses, but given that we perceive them as different colors, shouldn't you always have some sort of increase in activation, but that increase could, why isn't the whole graph just pushed up a bit?
Right, okay, I think I understand your question, and it comes down to this. Another way of asking your question is, doesn't this result, doesn't either your approach assume or this result demonstrate neural tuning or receptive field sizes of certain widths? So let me see if I can make that a little bit more concrete.
So much more sophisticatedly.
Yeah, no, but I think I understand what you're saying. Suppose you have, so here's the space of colors. So here's the space of possible colors, right? So remember we had that cube of colors. Okay, and so this is, red along this axis and green. Okay, so this is all the possible colors live within this cube. Okay, so now you might imagine you have a neuron, right, which has a receptive field which is tuned for just that color. It only likes that reddish-green color. Okay, and its receptive field might be so small that if I present a different color, this neuron doesn't respond at all, okay? But if the receptive field of the neuron was bigger, so it responds to many different colors, but it responds best to this color, then that would be a form of tuning which would be compatible with what we're measuring here, okay? For this to work, for this kind of result to happen, it would have to be the case that neurons have receptive fields within the color space that are large enough so that they can be strongly stimulated by one stimulus and have a bit of a weaker response to an adjacent stimulus. At least that's one model that's consistent with these results. Does that help answer that?
So it's a fully continuous space with peaks and valleys, I guess.
That's right. Well, so the argument would be that this result suggests that there's a population of neurons that are capable of representing the entire space of the colors with their population code. That there's some neurons that like greens a little bit more and some neurons that like reddish colors a little bit more and some that like purples a little bit more, and that combination gives this effect. It turns out, and actually as we're going to talk about in the second half of this lecture this afternoon, is that there's another way that you could get this result with neurons which have slightly different tuning, where you might have some neurons that just code for how green a stimulus is, and other neurons that code for just how red a stimulus is, and never actually explicitly code for the combination of green and red. You just have the combination of the two populations of neurons. But we'll save that for this afternoon. That's where we start talking about the shapes of receptive fields. Okay, so in any event, so you can measure this kind of adaptation, which is proportional similarity. I don't want to spend a long time on this, but it turns out you can measure these other kinds of carryover effects in these designs as well, things like bias. So this is a covariate that models, is there a bigger response to seeing a bright stimulus after a dark stimulus? Right? So if the colors go from the darker colors to the lighter colors, do you get a bigger response? And it turns out that within primary visual cortex, we get responses which are consistent with what's been seen before for what's called lightness adaptation or lightness gain control. So you get a modulation of the response to a color dependent upon whether you're going from dark to light or light to dark. Okay? So that's just another kind of carryover effect that's different from adaptation, but that can also be measured in this data. Okay. So up to this point, I've spoken only about neural adaptation, measurement of neural adaptation and carryover effects. But in this very same data set, you can also examine the direct effects of the stimuli and then look at the pattern across a set of voxels for like multi-voxel pattern analysis. And so the way that goes is that there were 27 times, right? There were many times that shade of brown was presented. And what you can do is you can get the average response across all presentations to that shade of brown for a given voxel. And you can do the same thing for a different color. That shade of green, every time it was presented, you get the average response across all of those presentations. And do that for another shade of whatever that color is. Okay? So you can do that for every different color. You get what the average response was to cross all those. And then this is important. Because the sequence is serially balanced, because it's counterbalanced in this way, this measure is mostly independent of first-order context. You're getting sort of what is the response to a stimulus regardless of what came before, right? Averaged over all possible things that could have come before, what's the average response of the stimulus, okay? And those average responses then can be used as the basis of a multi-voxel pattern analysis. You know, for those who like looking at covariate matrices that look like this is what the set of regressors or covariates look like to model that kind of data. You have one covariate that models all the time peach was on the screen, and all the time dark green was on the screen, and all the time blue was on the screen. Okay? So that's a modeling of each occurrence of that color across the entire thing. So you can then use the pattern of responses to those colors across the voxels to create a neural similarity matrix, right? So this is the kind of thing that Nikos Krigaskort has described and people do quite frequently now, which is where you say, here's my set of, in this case we took 1,000 voxels from within visual cortex. Here's the set of values across all those voxels for violet and for green, and you look at the correlation between those two, and then that comparison between just two colors is one cell of a neural similarity matrix. Okay? And you repeat this for every possible pairing of the colors, and you come up then with a neural similarity matrix, how similar one color was to another color. And then you compare this neural similarity matrix to how perceptually similar the colors were. And to make a long story short, there is some similarity. You can get a above chance correlation between the distributed pattern of neural similarity and the perceptual similarity of the set of colors. It turns out most of that similarity is being carried by just how the lightness of the stimuli and less the green and blue components. It's more how bright or dark the stimuli are seems to drive most of the across voxel similarity for the cortex. But just as a didactic point, this is a way that you can analyze the data in a different manner and come up with a measure of the direct effects, which can be used for multivoxel pattern analysis. Okay? So both sorts of effects exist within the data simultaneously. You can just analyze them one way or another way to see both of them. Okay? And this then affords you the opportunity, it gives you the chance to compare what the tuning or representation is like for within voxel adaptation and across voxel pattern analysis, which is what we're going to come to in a little bit. Okay? I'm going to give you one more example study, and then we're going to take a little break, and we'll come back and talk about some other things. So, okay. So population coding of face similarity. So let me show you an experiment then that does the same kind of thing, not with colors, but with faces. So the model that you might have to test this idea, oops, go back one. Okay, so the basic idea is that within a portion of the ventral cortex, there might be a population of neurons that represent the appearance of a face using a population code. Right? So you have a distributed set of neurons, or an intermixed set of neurons that the pattern of their firing represents the appearance of one face or another. Okay? And so just to be a little bit more concrete, a very simple cartoon model of what that circumstance might look like is that you have a space of different faces. You have these 4 different faces, and that you have a set of neurons. And the idea is that you might have one neuron that's tuned to respond best to that face, and another neuron that's tuned to respond best to that face, and so on and so on. So different neurons respond best to different faces. And if you present one particular face, then the response of the set of neurons is maximal for one neuron, and then some other neurons respond a little bit because the face is close to their receptive fields, and so on. Okay? And again, the idea is that adaptation can be used to measure this idea, the degree of response and the similarity. And basically, the adaptation is measuring how dissimilar 2 faces are within sort of this multidimensional or population code representation of faces. Okay? This idea that you might have faces being represented as points within a multidimensional space, right? That you have a set of neurons that code for the appearance of faces within multiple dimensions is one that's been around for a while, and it's appealing because it seems to account for some aspects of similarity that you might notice between different faces. So just for example, a pair of faces might be similar in some ways and dissimilar in others. So for example, here's a pair of faces where it's the same internal facial features, but it's a different skin tone and external appearance. This is actually a woman who was an actress on a TV show in the United States that was a very bad TV show and only lasted about a week before it got canceled. But they took one family, and they had the family did makeup so they looked African American, and they took another family that was African American and they did makeup so that they looked white, and then they went out and they filmed them while they did things. But this is just an example of how you can have faces that are similar along one axis but differ in a different way. You know, this can apply not just to faces of people, but animals can actually have this kind of similarity too. So these 2 things are similar in some ways but different in others, and this can even apply across all sorts of species. Poor Nick Nolte. So so there's this idea of similarity, right? Of the perceptual similarity of things can vary along different axes. This has been studied before with paired adaptation, right? And so this is a study by Zhang and colleagues in which they showed pairs of stimuli and showed that the recovery from adaptation was proportional to the dissimilarity of the faces in the space. I'm going to show you just real quickly an example of a similar kind of experiment done with this continuous approach. So here's a set of 27 faces that differ along 3 axes: identity, skin tone, and then the thickness of the face, whether it's a thin face or a thick face. And you can do behavioral measures to see how similar any one face is to another. So you have people look at pairs of the faces and make judgments about whether or not the faces are the same or different, and you use their behavior to determine how perceptually similar all the faces are to any others. Okay, so this is the similarity of any one of the 27 faces as compared to any other one of the 27 faces. Okay, and if you do that, you can then recreate a perceptual similarity space. So this is a representation of how similar one face is to another, right? So these two faces are relatively similar, and these two faces are relatively dissimilar as judged by our subjects. And so in an fMRI experiment, you present these faces one after the other. And what subjects were doing was that they were watching these faces, and any one face came in a slightly older and a slightly younger version. Okay, so that's the younger face, and that's the older face. So you can see it's a subtle difference between them. And so what the subject had to do was they had to judge, is the face the older face, or is the face the younger face? And so that's what they watched, but what we're interested in is what's the relationship between a face and the one that came before it. Okay? And so what we're going to do then is measure what is the neural response to a stimulus as a function of the similarity of the face that came previously. Okay, so again, this is an approach that should be familiar from the last example. You basically model for each face transition, were the faces similar or were they different, and then that models the adaptation. Okay? I will say actually that this experiment was designed for the purpose of looking at some additional, more complicated questions, which I won't discuss today, but that a student of mine, David Kahn, is presenting at the Society for Neuroscience in the fall. So in any event, if you take that linear adaptation effect and measure that response across a group of subjects, you find that there's a linear adaptation effect in ventral visual areas corresponding to the fusiform face area. So that works just fine. And you can do this same kind of experiment for a different set of faces. Now faces that vary in identity and race and their gender. Okay? So we have a set of faces that are synthetic, that vary in this way. And we can do the same thing. We sort of measure their similarity, their perceptual similarity. We create the similarity space. And then we analyze that data as well to look at this linear change in similarity. And it turns out that it's the exact same kind of pattern. You can measure the same sort of recovery from adaptation that's proportional to similarity. There are some very interesting additional questions to be asked about. So how does coding for gender changes versus identity versus race, how do those differ? But I'm not going to talk about those right now. That's for David Kahn to talk about at his neuroscience meeting coming up in the fall. The main point here is just to say, again, you can measure this linear similarity and measure adaptation that's proportional to how perceptually similar the faces are, and it works for, in the same way for two different sets of faces. Okay. This is the final thing I'll say and then a break. So what we've talked about so far is this idea of a population code that can represent the similarity of a set of things with a set of overlapping receptive fields. What becomes interesting is that if you take stimuli now, not along a single dimension, but along two dimensions, then there's a question to be asked about the tuning of the neurons within those two dimensions. Okay? And so here's the two possibilities that I'm going to offer, and then we'll talk about after the break. So one possibility is that you have a neuron which responds to a point in that set of faces, and it responds only when it's a particular face within that space, right? That it responds to a particular point on both axes. Another possibility is that you have a neuron that's tuned where it has an equal response along one axis and has a particular tuning along another axis. This would be a neuron, for example, that codes just for gender or just for the racial appearance of a face, right? So this was a face that might just respond to, to having faces which are male, but doesn't care about the identity of the face, right? So this is one possible tuning. Or you might have neurons which respond only to a particular identity and a particular gender. Okay? And so this possibility of a difference in tuning for just a particular point in a two-dimensional space or being equally responsive to stimuli along a given axis in the space is something that we'll return to after the break. So let me stop there and see if we have any questions about this piece before we come back for the second half. That's right. Okay.
For designing these carryover designs?
That's good. The question is, what do we assume about the shape of the hemodynamic response function for this? So the way that this is generally done is we assume an average population hemodynamic response function, but then also include a regressor which models any temporal shift of the response to the data. But that temporal shift only operates over short time scales, so it can model about a second or two of temporal shift either way. And that's sufficient to model the data accurately for all subjects.
And I guess it will also depend on the TR of the sequence, on the sampling rate of the sequence.
Well, we normally do these experiments with the timing of the stimuli is asynchronous to the TR. So the TR might be every 3 seconds, but the stimuli appear every 1600 milliseconds.
But the actual sequence will depend also on the TR. It is one of the parameters when you have to design the sequence, to compute the sequence with
right.
the program, because
right.
this is a program. You can do it.
Yeah, right.
So you have to input the TR, or
no, the TR does not enter into the calculation of what the ordering of the stimuli is that you need. The idea is that if you, it's just let
me say, like the sampling of the HRF, just the sake of the HRF, no?
No, because again, we're not. The property of this is that here. So if we come back to this example covariate. Here. So so here's this example covariate, which is created to model every transition, okay? This then gets convolved with the hemodynamic response function to model it, okay? So these tiny little ups and downs get wiped out once you convolve with the hemodynamic response function. And what you have is just the overall envelope of the rising and falling of the modulation over the course of the experiment, okay? So it's really just the lower temporal frequency component of this which carries through. So we're not actually recovering the precise neural response to each individual pair, but instead overall what the form of that modulation is across the entire experiment, right? Carried by this model. Does that make sense?
Yeah, and so when you include quadratic activators, it's because you are worried about or concerned about the nonlinear effects on the HRF or on the neuronal response.
Actually, on the neuronal response is what we're most interested in.
Linear system.
That's right. That's right. That's right. That's correct.
Okay.
Yes.
Given your starting point, your premises, I lost something. That is the brain, the spatial brain resolution of what you are doing here.
Mm-hmm.
Because at the end, it seems that you are finding modulations on the response in the specific brain regions, but at the end, it seems that you are not finding modulation in the geography of your response in the brain, depending on
right. That's right.
your dimensions.
No, you're correct. This is just the setup. So what I've described now is just to convince you, or is to understand the method and to convince you that we can measure a neural modulation that is proportional to similarity. And so now the interesting work can begin, which is to ask, how does neural tuning vary for different stimulus types, and how does it vary across spatial locations, and all the rest. And so that's what's coming up in the second hour. Yeah, no, of course. No, but that's fair enough. No, this is just the prelude to then actually then applying this to questions of neuroscientific interest coming up next. Okay? All right, let's take a break. Still here in this room because I don't think I could listen to anybody talk for one person talk for this long and actually be able to stand it. Heck, I love my wife dearly, but I think if I heard 4 hours, 2 hours a day for 4 days of just lectures from her, I don't think I could do it. So again, thank you very much for your attention and hanging in for all of this. And tomorrow has the property that will be a very different topic and a very different set of ideas. So at least, even if the person is stale, the ideas might be fresh. So, okay. What's that? So now I'd like to talk about an application then of these ideas of carryover designs to looking at a really basic psychological property of perception. And this goes back to, again, like the 1950s of psychology and an area called configural processing. Configural processing. This is really neat. These are some very fundamental ideas about how it is we perceive the relationship between different ways that things can vary. So here's the basic idea. So let me see how good the colors are for you guys. Well, that's pretty good. Yeah. So, okay. So the key insight is the following, and that is that you can have a set of things that vary, and for some variations, you automatically and effortlessly perceive the variations as different things, and for other sorts of variations, you don't see them as different things. So for example, color is the classic example of what's referred to as an integral space or an integral perceptual space. And what that means is that if I just took a naive observer and I showed them this and I said, what's different between those two things? They would say, the color. The color's different. They'd be very unlikely to say the hue has changed and the brightness has changed. Only if you specifically prompted them to be sensitive to that dimension would they say that. They'd just say the color has changed. In contrast, if I showed this thing and said what's different, they'd say, well, the color has changed and the shape has changed. Okay? So these two dimensions of the space are an example of integral dimensions. You don't automatically see them as different things. Whereas these two dimensions are what are called separable. You automatically and effortlessly perceive them as being two different properties of the set of objects contained. Okay? So those go by the names of integral and separable. Work going all the way back to Roger Shepard showed that within spaces like this, the similarity of stimuli uses a different metric, or it can be measured as a different metric. So within an integral space, the similarity of any two stimuli is just their direct distance within the space. That is, the distance from this stimulus to this stimulus is just the straight line distance between them within the space. That's how perceptually similar they are, can be defined as just their direct distance. Okay? But within a separable space, the perceptual similarity of any two stimuli follows what's called a city block metric. Okay? And the idea is that the similarity of these two things is 1, 2, 3, 4 steps away. Okay? Whereas the similarity of these two guys is whatever the hypotenuse of 2 and 1 is. Okay? That's sort of a basic observation about the perceptual similarity of stimuli that are drawn from integral and separable spaces. And again, this is an observation that goes back for many years in psychology. And this kind of thing has been studied in many different perceptual domains. In color, there's a lot of really great work that I just had a chance to discuss briefly over the break in the perception of sound. So the relationship between the pitch of a sound and its timbre or timbre, I don't know how to say that word, and its loudness. So just how different dimensions of sound variation, for example, are either perceived as integrally or separably, properties of objects, and then so on and so forth. Okay, so there's a rich and somewhat old literature in how different spaces have these properties. And what I want to offer for us today is a neural mechanism that might correspond to when some stimuli are seen as integral and some stimuli are seen as separable. So that's the goal. So now, practically, the way that integral and separable representation is measured is using something called Garner Interference. So this is the technique that's used. And essentially, it amounts to this. You present subjects with stimuli from the space under two different circumstances. In one circumstance, you say, what I want you to do is tell me, is it the 4-pointed star or the 7-pointed star, 2, 6, 7, 8, 7-pointed star. So you have to say 4-pointed or 7-pointed, but the stimuli come in different colors. Okay? So you have to say So you might be shown that stimulus, and you have to say it's 4-pointed, or that one, and you have to say it's 7-pointed. Okay, so you judge the shape and ignore the color. In another condition of the experiment, you are only ever presented with the 4-pointed red and the 7-pointed yellow, and you still need to judge the shape. So you have to say that's 4-pointed, that's 7-pointed, but there's never a variation in color. Okay? And the idea is that you need to make this judgment, you need to ignore the color when you assign the label of 4 or 7 points to the star. And just as you might be able to introspect, it's actually trivially easy to judge the shape and ignore the color. You have no problem whatsoever where you say 4 or 7, 4 or 7, 4 or 7. You don't care about the color at all. You're just as quick to make that judgment, right, when the color can vary, as to make this judgment when the color is fixed. Okay? So this is the reaction time across 9 subjects for making that judgment for when the color is fixed and when the color can vary. Right? So this is the labels will come up in a moment, but I think this is when the color varies, and that's when the color is fixed. Contrast that now to making this judgment. Now you need to make a judgment about the hue of the stimulus, or I guess in this case it's brightness, while hue can vary, right? So these are darker and these are lighter, but they also come in more yellow and more red. These two don't look very different on your screen. So you have to judge darker or lighter and ignore yellow or red, and either do that with it's either they can vary or they don't vary, and now there is a lot of interference. Okay? It's much harder, or you're slower, to judge darker or lighter when the stimulus can come in red or yellow than when it does not have that variation. And so that difference is what's referred to as Garner interference. It's the interference of the ignored axis upon your perceptual judgments. Okay? Now, so that's taken as a measure of integral or separable perception, right? So for these spaces, because there's no interference, they're seen as separable dimensions, hue and shape, whereas since there is interference, these are seen as integral dimensions. I should say that these are not absolute categories. So Garner interference can vary continuously between different stimulus spaces. Here's just some examples from some stimuli that we tested in the lab. So shape and color, that's the as a percentage of modulation or percent of interference on reaction time. That's how much shape and color interferes, so not much at all. But then if you have to judge, for example, so these are stimuli in which just the eyes can change or just the mouth can change, and you need to focus on just the eyes or just the mouth. It's actually possible to make a judgment and focus just on one face part and ignore the other parts. We'll come back to this a little bit later on. Here's some shapes where you have to judge curvature and their thickness, and so this is a little bit more interference. Here's a case where you need to make judgments about the texture of a stimulus and ignore the scale. So whether or not it's sort of high spatial frequency or low spatial frequency, more interference. Making judgments about whole faces. So this is where these faces now vary in like a holistic identity along one axis or another, and it's very hard to put people into this category or this category while ignoring the other way that the faces can vary. And then finally, the most integral of stimuli that we've been able to find in the lab so far are these popcorn shapes, which I'll tell you more about a little bit, I think, tomorrow, or a bit more today, where these are now shapes that vary along 2 dimensions, but it's hard to even see what the dimensions are. I mean, they have a strict definition in terms of their mathematical creation, but you just see them as different shapes. It's very hard to perceive that there are these 2 dimensions along which they're arranged. Okay? Okay, so here's the hypothesis then. Here's the idea that I want to motivate the study I'm going to show you, and that is that integral spaces are represented by conjointly tuned neurons. That's the basic idea. So if you find as 2 dimensions, like hue and brightness of color, where people can't tell them apart very easily at all, then what we want to posit is that you will find neurons which are tuned to respond to a particular point in the space with a circular receptive field. Okay? That is, you'll have one neuron which happens to like this particular color. It needs a particular combination of hue and brightness to fire. And there's another neuron that liked this particular color. It needs a dark orange whereas another neuron likes a bright yellow. Okay? So conjointly tuned neurons will be associated with integral spaces. In contrast, where you find separable spaces, you'll find neurons which are independently tuned. That is, you'll find some neurons which code for a particular shape independent of color, and other neurons which code for a particular color independent of shape. In fact, that shouldn't be such a surprising or controversial claim, because we already would have the suspicion from what we know of the way that ventral occipital cortex is organized that you will find neural populations like this. Color responsive neurons that don't care about shape, and shape responsive neurons that don't care about color. Okay? But it's going to be, as we'll go along, within a localized stimulus space, the relationship between conjoint and independent neural tuning that's of interest to us. So what I'd like to show you is that there's a way that you can use these carryover designs to basically measure this property, to test if neurons are conjointly or independently tuned for a particular perceptual space. So here's how that works. Okay, so there's a space of stimuli, and now let's imagine one voxel, right, that's coming from somewhere in the ventral cortex. And within that voxel, we're going to have two possible cartoon models, or two extremes. So one extreme is that it contains neurons which are conjointly tuned, and the other extreme is that it contains neurons which are independently tuned, right? So neurons which code shape but ignore color, and intermixed in that voxel are neurons that code color, not shape. By the way, it's trivially easy to know if you had different regions of the brain, one that liked color and one that coded for shape. That's actually a trivially easy thing to find. What's hard is if those two independently tuned populations are intermixed in a single voxel. Okay, and so that's the case that we're going to focus on here. It turns out that there's a way that you could measure the difference between these two cases, and it's like this. Imagine a transition between two stimuli in the space. So a transition between that stimulus and that stimulus. What I'm going to argue is that the neural dissimilarity, so the measured dissimilarity that the neurons represent, is city block, just like the perception. That is, it's the sum of the dissimilarity along one axis, that's one population of neurons that are coding for shape, and the dissimilarity along another axis, that's the population of neurons that are coding for color, right? In contrast, if the neurons are conjointly tuned, the population similarity is instead the dissimilarity that's related to a Euclidean metric, how far apart they are as the crow flies. In practice, what this looks like is, if you had a stream of stimuli coming by, you'd have two covariates. One covariate that models for each transition how much of a change there was in color, and for each transition, how much of a change there was for shape. And these two covariates are all you need to model the recovery from adaptation for these two populations of neurons, right? So if you had neurons, some which cared about shape but didn't care about color, and some which cared about color and didn't care about shape, and they were intermixed in a voxel, these two covariates could model the recovery from adaptation for this set, right? However, if the neurons were conjointly tuned, you would have a smaller recovery from adaptation, a smaller response for a combined change, right? A diagonal change in the space. You'd get a smaller response than would be predicted by these two, okay? And so a linearization of that we refer to as a Euclidean contraction. It's basically, it measures how much smaller the response is for diagonal movements in the space than you would predict based upon the amount of recovery of response you get for pure changes along the axes. Okay? Let me stop there for a second and see if there's any questions about that set of ideas. Does that seem perfectly clear? Yeah.
At the end, what you're claiming is that you can have two streams of adaptation in one case and only one in the other.
Two streams of adaptation?
Yeah, two dimensions of adaptation in one case, 1 and 2.
Right.
And in the second you have just one.
That's right. Exactly. That's exactly right. Yeah. So as one other way of doing this would be to analyze the data with two different models. one model where you use two covariates, right? This two-stream model, and the other analysis where you use a single covariate, and then you compare the goodness of fit of the two models. That would be another way of doing it. We have, as you'll see, we have reasons for wanting to quantify the degree of this Euclidean contraction because it turns out that life is more complicated than purely conjointly tuned and purely independent. There might be tuning that's intermediate, and this allows us to quantify that. Yes?
Just, it doesn't have to do with the hypothesis that you're showing, but do you consider that those mixed conjoined neurons are just integration, neurons that make integration, or like it's not that the neuron codes for shape and for hue, but those are neurons that depend on the other neurons.
I see. Okay. No, I got it. So.
There's nothing to do with the hypothesis.
Right. So you're saying that it could be the case that you would have a neuron which integrates the firing of a lower level. That's right. Though I would say that for the purposes of this model, I would say that's still a neuron which responds to the joint position within the space. That's right. That's right. So, okay. So to do this with functional MRI and within this carryover approach, there are a couple of optimizations that must be done. So here's one optimization. So far we've been talking about spaces, a set of stimuli that are from a square grid. It turns out this isn't the best way to examine these ideas. It's better to have stimuli which have a lot of diagonals, okay? Because the diagonals are the most informative transitions, right? The transition from here to here along a diagonal versus a pure change. And so this sampling of stimuli from a space is the most efficient for being able to test this idea of conjoint versus independent tuning. You want to have a roughly equivalent number of pure changes and diagonal changes. And so this kind of sampling from the space is an optimization. I will say that in the paper that we wrote that describes this technique, we spend a lot of time doing simulations to show that Euclidean contraction, right, that measure of conjoint or independent tuning is robust to nonlinearities in either the spacing of the stimuli or in the hemodynamic transform. So without getting into all the messy details, we spend a lot of time modeling neural populations and considering various nonlinearities in either the perceptual representation or in the hemodynamic transform. And so those nonlinearities, we examined whether or not that could fool us into thinking that a stimulus was conjoint or independently tuned when it wasn't really. And to make a long story short, there's only a couple of specific instances where the approach fails, and we have ways to test for it. I won't say any more about it, but anyone who wants to read the appendix with me, I'll be happy to go over it with you. But it's very robust. It's very robust to every plausible nonlinearity of the system that we could find. There's one other thing. There's one other way that this test might get fooled. And that is, what if you picked the wrong dimensions, right? So what if you thought that the tuning was for color and shape, but really the neurons are oriented to tune for some combination? That is, they still have these independent receptive fields. It's just you got the axes wrong, right? You thought it was color and shape, but it's actually something a little different. So it's a rotation of the axes within the perceptual space. For color and shape, that doesn't seem very plausible. But for other dimensions, it could be the case. So what we show is that there's a test you can do upon the data where you basically you rotate the covariates. You ask if you modeled those covariates for a rotation of the space, does that Euclidean contraction go away? And the basic idea is that for conjoint tuning, for when you have neurons with circular receptive fields, it doesn't matter how you rotate the covariates, you always get the same result. But for when you have independent tuning, it drops and comes back. So I won't say again too much more about this, except we have a way of testing to see if it's actually an independent space, but we're just rotated from where it should be.
For this N,
Oh, let me see if I can remember now. It is the number of stimuli that it's the number of stimuli that make up the space. It's how densely we sample the space. So that's right. Oh, thanks. Good. Yeah, good. Thank you. Yeah, good. You read along with me and let me know. That's right. It's basically showing that that even for if you have a coarse sampling of the space, you can still detect this effect. It's a nuance. Okay. Mainly the point of this slide is just to say that loading on the Euclidean contraction can tell us not just absolutely is it all the way like this or all the way like this, but also identify the intermediate case. Okay? So, and there's a linearization of this so that we can measure this Euclidean contraction and find, are we more at this case or more at this case or somewhere in between? So we can actually map out where along that dimension we are. Okay, so how conjointly or independently tuned are the neurons? So one application of this that we've tried is for the integral and separable perception of space, of two-dimensional closed contours. So those shapes that I showed before, they come from a particular source. So Hans Optebeek, back in 2003, showed that with some behavioral studies, that you can create sets of shapes where the dimensions are either integral or they're separable. And the idea is that some of the separable dimensions, some of the dimensions that you easily perceive about these simple shapes, correspond to things like aspect ratio and curvature and thickness, very basic object properties. Okay? And those dimensions, those properties of shapes, seem to be perceived in a separable manner, whereas there are other dimensions of shapes which are perceived in a more integral manner. And this is something that we replicated and showed was the case with Garner interference. That is, it's harder to judge, this categorization of the shapes when they vary along these dimensions, which just don't look like anything to us, than it is to vary these dimensions. So here, you have to judge the curvature and ignore the thickness, and that you're much better able to do than to somehow group these two things and these two things. Okay? So that's just a measure of Garner interference, demonstrating that these shapes have this property of being more integrally or separably perceived. Okay? So then, what we wanted to do was test our hypothesis then that these integral dimensions of the shape space correspond to conjoint neural tuning within ventral visual areas, within shape-responsive visual areas, whereas these dimensions of the space are coded by more separable, independently tuned neurons. That there's neurons that care about curvature and don't care about thickness, and neurons that care about thickness and don't care about curvature. Okay? And so the way we did this was within one of these carryover designs. The stimuli came flashing on by, and the task that the subject did was that there was a line that went behind the shape, and the line could move and it would be moved or tilted in different trials. And what they had to judge was more of the mass of the object to the left or to the right of the line. So this forced subjects to look carefully at the shape and make a judgment about its shape properties without making an explicit comparison to the stimulus that came before. Okay? So this is the manipulation that we had in the scanner to get people to pay close attention to the shape. All right? So the first thing is that we were able to show that within ventral visual areas, well, that's an old picture of the brain, but within ventral visual areas, there was a recovery of adaptation for all four of the stimulus dimensions. That is, variation of dimension 1 and dimension 2 of these what we call popcorn shapes, and variation of dimension 1 and dimension 2 of what we call the moons. All of that produces a recovery from adaptation with those city block covariates. Okay? But the key question then is, in these ventral visual areas, and this mostly corresponds to HV4 and a little bit of V3, is there loading on that Euclidean contraction? Is it the case that a diagonal change within the space for the popcorn is smaller than would be predicted by the sum of these two sides? Right? That Euclidean contraction would suggest then circular receptive fields. And that's in fact what we found. So within these areas of ventral cortex that showed recovery from adaptation for shape variation, there was also an additional smaller response for a diagonal change in the space, so suggesting that the tuning of those neurons is circular within that space. In contrast, for the moons, you can see where this is going. We can measure to ask, is there a contraction? And the answer is no. There's no contraction between them. Okay? So what that would suggest then is that within this ventral visual area, you have circular receptive field tuning for these ineffable dimensions of shapes, but for the moon-shaped objects, it's more independently tuned. Okay? And because it's an important part of this, we also had to test to make sure for this rotation effect, and we could show that there's a slight modulation, but that it's a Euclidean contraction regardless of the orientation of the axes. So let me pause there for a moment and see if there's any questions about this set of ideas.
Yeah, George, I would ask you something about this complication on the spaces, because you told that in Euclidean space, okay, we can quantify this distance, but when you put another, the shape and the color you have, I don't know how the shape you can quantificate. You have different shapes, but in color you can have one difference from one to the next color. But
good.
With shapes, how do you quantify this distance to?
Definitely, right. So how do we know this spacing? Right? How do you know this? And the answer is multidimensional scaling on behavioral data. So you take subjects and you show them pairs of the stimuli, and you have them make a same or different judgment, and you measure their reaction time, and then you take those numbers and you do multidimensional scaling. And that's the classic technique for judging perceptual similarity using stimuli like this. That's right. So we did those tests to make sure that the stimuli were equally spaced.
Yeah, but I thought that there wasn't an global scale. That's perceptual.
That's right. That's right. Okay.
And what about how many samples can you define or can you think about in the space?
What's the limit?
Because perhaps, the Euclidean is great, but you cannot think of a shape that lies in the middle of your space.
Can't think of a step
that you cannot think, because you need to. I'm thinking about rotations or decision, and there are jumps, okay, that you cannot think of a shape that is in some particular combination, and there could be a possibility that.
Oh, what if there are holes in your space?
Yes, exactly.
Okay.
So that it's more difficult for you because you
that's right.
Think of
so
something which is there.
Okay, that's right. So I will say that this technique in particular for looking at this integral separable requires a stimulus space that has a fairly continuous instantiation. If there's a lot of holes, then your ability to test some of these things in a rigorous manner will be impaired. But in general, for doing these sorts of carryover designs and looking at similarity, it's okay to have holes. You could have any set of stimuli as long as you know their mutual similarity. You could then examine what's the relationship between their perceptual similarity and their neural response. Does that address your question a little bit?
If you want to go very fine details, is where the holes are becoming really important.
Yeah, I think that probably is right. So just in, let's see how we're doing on time. I think we're just about done. So just in the final moments, I'd like to just show you another application of this idea. By the way, for those who are interested, on Thursday afternoon, we'll talk about an application of this set of ideas to looking at integral and separable representation of face holes and parts. Thursday morning, we'll be talking about integral and separable holes and parts. For those who are interested in face perception, be these ideas. But for now, I just want to show one last example of this Euclidean contraction approach. And this is in a study that a student named Wesley Kerr conducted in my lab, and that we've just finished now, where we're looking at the relationship between texture and scale. So objects can come in different kinds of textures, right? So it's all teapots, but they have different kinds of surface properties to them. And there have been some studies that have looked to see how variation in texture identity is represented. And in general, this is a study from Kant and Goodale. They found that there is a region of close to area HV4 and ventral cortex, which seems to represent texture properties. But we wanted to look at something a little bit more subtle, and that is it the case that you have a representation of texture that is independent of the scale of the texture? That is, do you have a scale invariant representation of texture? So to do this, we needed to create a set of textures that had a regular spacing and set of properties. So we did this by using something called steerable pyramids and texture synthesis. We started with wood grain and stucco pictures, and then we used a technique which Eero Simoncelli developed, which is referred to as a steerable pyramid way of creating stuff. We created basically synthetic wood and synthetic stucco. And the reason we did this is because we can then normalize a lot of properties of the stimuli and then create other stimuli that are intermediate. Okay? So using this synthetic approach, we can create 2 dimensions of stimuli. So stimuli that vary in texture identity, from this is wood, and this is stucco, and this is something in between. And stimuli that vary in spatial frequency. This is fine-grained, and that's coarser-grained. Okay? And we can do, again, like this multidimensional scaling technique to be sure that behaviorally the stimuli are equally spaced. So we do those things. And then instead of sampling the stimuli from a grid, we instead grab the octagonal arrangement of the stimuli, so they look more like this. So now we have the stimuli sampled from this sort of octagonal arrangement. And there's a further component, which is that because we're synthesizing the stimuli, we can create different exemplars of a stimulus from a point in the space. So this stimulus point here is, a certain amount of stucco and at a certain spatial frequency, but we can create different exemplars of it. Okay? So during the scan, people see different pictures. They're actually individually different pictures, but the pictures have the common property of being at a point in the space. And so we must be ready to wrap up. So we can do this for different sorts of textures and then run a carryover design just as you've seen, where the stimuli come on by and we vary the exemplar that the subject sees. And just as we've talked about before, you can create a stimulus that you can create a model of the change in texture identity and a change in texture scale. And so if you look across the entire brain at where there's a main effect of seeing stimuli versus the blank screen, this is all back in ventral visual areas. But moreover, we can look at what the adaptation effect is for texture and scale. So this is the sum of the two of them. This is a patch of visual cortex, visual area 1, 2, 3, HV4. So this is where there's a recovery from adaptation, which is proportional to the change in texture and the change in scale. And remember, the stimuli are actually different on every trial. They're never the identical stimuli. They're from just the same place within the stimulus space. And we can show that as you move from early visual areas to later visual areas, you develop a more scale invariant representation of the texture. So the basic idea is that within HV4, you start to have a representation which is more of texture and less of scale, although it never becomes totally invariant in the measures that we obtained. Finally, in data like this, you can examine whether or not within those regions you find independently or conjointly tuned neurons for this kind of representation. Use the same kinds of techniques that we've talked before about having a Euclidean contraction for this, right? So, but here's the thing. Behaviorally, if you look at the relationship between these two axes, if you test a bunch of subjects, it turns out there's a lot of individual differences. So we did this using not Garner interference, but a similar kind of technique that's called an Ashby-Maddox measure for integral or separable perception. And we used it because it has a lot of test-retest reliability in subjects. So we can measure a subject's tendency to see the stimuli as integral or separable. And so we obtained that measure for a group of subjects, and we also obtained the Euclidean contraction, so how conjointly or independently tuned the neurons were. And across our group of subjects, there's a relationship between those two things. It's a small number of subjects, so I don't want to make a huge deal about it. But it is the case that the more integral you see the stimuli, as subject sees the stimuli, then the more conjointly tuned the neurons appear to be by this measure. So that's where we're taking this kind of work to try and take a look at individual differences in perception and relate them to neural tuning. You can also imagine how you could take subjects who perceive stimuli a certain way, train them to now see stimuli as separable, and then test to see if that's altered the neural tuning. So I'll stop there. Any final questions before they kick us out? Yeah?
Yes. Are you aware yet about any kind of studies that have been trying to look at this neural adaptation in terms of functional connectivity? I mean, like the Jesse Risman method, like, you know, areas that are firing together in time, see if that neural adaptation is simultaneous for both areas or...
Okay, that's interesting. So to see if there are... Well, here's what's, so you'd have to look, the question is about resting state connectivity and if that could add something to this. It'd be hard. You'd have to believe that you can get a component of the fMRI signal that's not being driven by your task, right? And then look at the correlation between the two regions in that signal. The problem is for this kind of design, I don't think I'd ever be confident that any particular component of the signal couldn't be attributable to some aspect of the task, because the task produces power at many different frequencies, and it would be hard to do. But nonetheless, it's interesting.
The most simple task, I mean, a very, very simple task, like, I don't know, even a finger tapping, or I don't know. I mean, that finger tapping would not be possible to do, but yeah, that was one thing.
Yeah, no, it would be, well, so that's right. It might be interesting if you, for example, if you found different cortical regions that were independently tuned, to then know, is there different connectivity between those regions when you're making judgments which have to combine shape and color and things like that. Yeah.
Which links to the question is that, are there any work that tries to do experimental design considering that the test is about functional connectivity?
Okay.
In terms of localization of the areas, but you want to test functional connectivity between areas.
So
even more directionality between areas.
So I am skeptical of the ability of fMRI to test directionality, but I'm not alone on that. Or there are those who disagree with me, who think you can use fMRI to test directionality. Yeah, that's an interesting question. Are there, I think that someone who's looked at this kind of thing is Christian Buchel. So he's conducted studies in which he has had variations in the task, used variations in the task, and then examined if the connections between brain regions are altered as a consequence of that. I'd have to think a little bit about exactly what would be good experimental design for those kinds of questions. I haven't really thought about it before. That would be kind of neat.
For designing this kind of experience in visual perception, is it important to select people with no previous experience with this kind of stimuli, or it does not make a difference?
So yeah, the question is, should the subjects not have seen the stimuli before? I think that actually is, that's an interesting feature of a scientific question, right? So what does familiarity with the stimuli do to these sorts of measures? So for the examples I've shown you before, they're novel stimuli. They're things that the people have not seen before. But one might be interested to know what happens to the representations as a function of training or experience or familiarity. I mentioned to somebody over the break that we are using these sorts of techniques to look at much more subtle aspects of color tuning. So, I started off with an example of color. Well, we're quite interested in if you have a one dimension of blue to green, for example, and all these colors you'd call blue and all these colors you'd call green, but the variation is continuous. So then how does visual cortex respond to that continuous change? Does it have a categorical response to blue to green, or does it have a continuous response? And the answer is a bit of a mix of both, which can then further be influenced by the task that the subject is doing. So I think that the familiarity with the stimuli, the task that someone's doing, all of these are the raw material of interesting questions to be asked using techniques like this. All right, well, I think we're over time. I'd be happy to answer more questions. And guys, thanks again for your attention.